Okay, so last time we talked a bit about how proteins are made, and I got to the part where tRNA brings in amino acids. And I said that amino acids are the building blocks of your protein. Now, one of the things that I should mention is the process from going from DNA to RNA is known as transcription. Transcription is when a part of your DNA gets replicated or transcripted into an mRNA using RNA polymerase, which is your enzyme. Now, from that process, we brought it into the cytoplasm, where the mRNA binds to a ribosome, which then brings in tRNAs, which read sets of 3 nucleotides, which are called codons, and that starts your protein sequence. This process is known as translation, which makes sense because if you have an encoded sequence that you want to translate into something useful. The mRNA in itself is not useful. It doesn't do anything besides code for a protein. These proteins later on do functions within your body. For example, hemoglobin is an example of a quaternary protein, which I will get into these different classes of proteins later on. But it's necessary for your blood. It cleans your blood. Another protein would be other steroids. For example, cholesterol is a steroid. That's also necessary for your body, so your arteries don't collapse in themselves, and they repair your arteries. Now, so we went through translation, and what happens is the ribosome, or the tRNA, keeps coming in with new amino acids as it's reading the mRNA, and it enters into your A site, which is known as your acetyl site, or your acyl site, sorry. And then in the middle, you have your P site, which is your peptidyl site, which is where the peptide bonds, or the bonds that connect your amino acids to produce your protein, are made. And then you have your E site, which is your exit site. This is where the tRNA goes, where it gets kicked out once it's empty. Now, once your protein is made, it gets taken to the place where it's necessary. It could be useful anywhere, pretty much. The cytoplasm, outside of your cell, going into a different cell, it can be anywhere. So like I was saying, there's different types of proteins, or different classes of proteins. First are your primary. And what your primary are is they're just your original amino acid sequence. Once it leaves here, right, that's your primary protein. It doesn't have any function yet. It needs to be folded. And then you have your secondary. There's 2 types of secondary structures that you can have. So this is your amino acid. Amino acid sequence, and then your secondary, you can have 2 types. You have beta pleated sheets, and then you have alpha helices. Now, the alpha helices look similar to your DNA structure, so it's going to be anti-parallel, and then you're going to have your beta pleated sheets, which just look like pieces of paper. And what these are is they're your folding techniques. Now you have what we know as motifs. So what motifs are is when an alpha and beta pleated sheet come together and they form different structures. For example, you can have an alpha-alpha, and then you can have a beta-alpha, and you can have all these different types. And then what you have is your tertiary. And what your tertiary are is just your motifs interacting with each other to make bigger proteins. And then finally, you have what we know as quaternary. What these quaternary ones are, this doesn't have to be done actually. Tertiary is where you have a functional protein. And when it gets to quaternary, that's just your tertiary proteins coming together to produce a larger one. For example, when I mentioned hemoglobin, Hemoglobin is made up of 4 different quadrants. It has 2 beta subunits and 2 alpha subunits. And this makes your hemoglobin act the way that it does. And what happens is with hemoglobin, you have an alpha one here and a beta one here, and then you have a beta one here and an alpha here. And basically, depending on the orientation of these, it determines what the function of your protein will be and what kind of protein it will be. Subscribe to me so you guys can see some cool videos doing science and experiments and maybe learning something new. Thanks!